Okay, perfect. All right, thanks for the introduction, and it's my pleasure to share with the groups here, members, I think a really timely and disruptive technology that allows for rapid high-throughput screening for vitamin D status, which is really a global health concern. Tragically, vitamin D deficiency is widespread, and it's significantly undertreated, underdiagnosed, and underprevented. So the main problem, as I mentioned, is the high prevalence of vitamin D deficiency globally. Upwards of 75% of the population, certainly in countries like Canada in winter season, are deficient or insufficient below a concentration of about 75 nanomolar, which is physiologically relevant for immune function in particular. It's a modifiable risk factor, so we can prevent that through proper nutrition, so sensible sun exposure as well. And it's associated with a plethora of sort of chronic diseases, including bone health, chronic pain, depression, cardiovascular disease risk, and importantly, I want to stress immune function. And this includes both cancer, autoimmune disorders, and of course, probably on the radar these days, COVID-19 and other respiratory illnesses. So the current methodologies for vitamin D screening and why it's maybe undersubscribed are methods that are available, but they're not so convenient for especially large scale sort of analysis and screening that are cost effective. This includes immunoassays, which are generally inaccurate, unreliable, although very convenient to perform, or instrumental methods based on LC10 to mass spec, which are more reliable, yet slower and higher cost for routine analysis. So our technology essentially bridges the benefits of both technologies, allows for rapid high throughput screening, but is more reliable than immunoassays and allows for high throughput screening. Generally, our method is a protocol that involves a series of steps, including a novel reagent that allows for chemical differentization and for separation-free based analysis that we can acquire data within less than 3 minutes per sample. So we can run hundreds of samples per day. And more importantly, on the screen on the right, the image, you can see we can get a dose response. We can actually accurately quantify the vitamin D status as measured by the blood levels of 25-hydroxy vitamin D3. That's the main storage form of vitamin D in circulation. So the main advantages of our methodology, again, it helps address, I think, a gap between these two existing technologies. It's faster and less costly than the existing gold standard method called LC10 to mass spec, and it has good mutual agreement with that. We've been able to rigorously validate the analytical merits of our method. And compared to the immunoassays that are also more widespread in use, it's more precise and robust and reliable, and it's not prone to batch effects. So we actually validated our method rigorously and actually compared it to a cohort of actually admitted children in the intensive care unit at McMaster Children's Hospital. This was a cohort of 30 children. We were screening them for vitamin D deficiency, and we did a three-way comparison between our method, the direct infusion 10 to mass spec, compared to the immunoassay or the standard method based on LCMS. You can see our method in the blue has less bias compared to the immunoassay, more than 2.5 times fold. And why that's really important is if you look at that population cohort, if we just relied on the immunoassay, only 16% would be kind of designated as deficient in vitamin D, less than 50 nanomolar, whereas with our method, we actually, and that essentially undertreats and underestimates the prevalence of vitamin D deficiency because it's less, again, accurate compared to our method. So we have accurate measurements to assess vitamin D deficiency for proper treatment. What's really exciting, especially during COVID-19 now, is high quality evidence that vitamin D insufficiency or early treatment with adequate intake can prevent respiratory illness, infections, hospitalization, and mortality ultimately. And this was a recent paper I really highlight if you're interested that was just published in Scientific Reports that reported of about 600,000 US veterans, overall 33% lower risk for COVID-19 related mortality with adequate vitamin D status that was prescribed in this particular case. So it's really on the radar in terms of the importance of vitamin D for immune health. But optimal dosage is required. There's a lot of variability in terms of treatment responses, which could benefit from periodic monitoring through an accurate, reliable method for screening. The market for vitamin D, both in terms of therapies or supplements, is increasing rapidly, especially again for the past few years. It's estimated to more than 5 billion in the next several years. And I think it comprises really a panel of metabolites and compounds we wish to measure routinely on a population level that can give us a good assessment of immune health. That again, it can empower individuals to take care of their own health through, again, dietary or lifestyle changes. And I think this is really an entry into the personalized health and wellness that could be done on a subscription type basis for routine periodic monitoring. So the big advantages of our methodologies, we just published our paper that attracted some media attention. As I mentioned before, we're a participant in the ongoing clinical trial of our essay in terms of assessing vitamin D deficiency and the effect or the treatment benefit of high-dose vitamin D supplementation in these critically ill children. Our main goal is, can we get away from the hospital setting and actually allow it for just a general sort of population use as well? Right now, we're evaluating the utility of blood microcollection devices, such as the ones on the right side that allow for home testing, either through a finger prick lancet sort of collection of a small amounts of drops of blood, or a device that can actually use on your upper arm for convenient collection as well. Look at stability, cost, and how we can use that as a very simple way to avoid individuals going to the phlebotomist for a large blood draw, which I think is a big barrier for routine testing overall. And the main idea you can see on the left side is we want to be able to first evaluate one's baseline status of vitamin D status, and then with follow-up monitoring with suggested sort of supplementation regimes or lifestyle modifications, well, you can get into that green optimal zone, which again is largely deficient for most of the population that can ensure, again, optimal immune health. So I think there's a lot of interest with, again, personal health and wellness for optimal nutrition. So the current status right now, we have a provisional US patent in addition to the published paper. I think we'd like to have a user-friendly, secure app that can provide, again, feedback, not just a diagnosis or where you stand in terms of your vitamin D status, but actually actionable items that people can take advantage of, and then have ways to validate whether the intervention or supplementation is actually effective or not. Besides vitamin D, we actually have a panel of other markers that are underway that are very interesting, that go hand in hand with inflammation and immune health, including omega-3 fatty acids, which is also a big sort of supplement sort of regime in terms of fish oil, calcium, magnesium, which are also kind of critical and often deficient in the population, and as well as iodine, which is a critical micronutrient as well. And the whole idea in our work is prevention, primary prevention of cardiometabolic or immune sort of dysfunction. But also we're taking part in a large-scale epidemiological study to look at other predictive markers of clinical events and outcomes. So this could be, again, a stepping stone to other types of applications. So our expertise is certainly in bioanalytical chemistry. It's perhaps not in business, as was mentioned by David. Assay development, rigorous sort of validation. We have a great sort of panel of collaborators in critical care, as well as in epidemiology. And we're really focused on nutritional assessment. So right now, I think we're poised to kind of translate the method and then go to maybe forming a startup company with the right partners as well. Again, the idea being personalized health and wellness. Our stage one is immune health, which will include, of course, the vitamin D assessment, maybe the omega-3 fatty acids and other additional panels. We're envisioning a second step in terms of cardiometabolic health as well, but ways to empower individuals to, again, modulate and improve their health on an individual level. Yeah, and I think the epidemic of particular concern is really poor metabolic health. And I think our current medical system is not really geared towards prevention, primary prevention, which I think this is the thing we want to focus on. And yeah, I'm very interested in open-minded to kind of partner with entrepreneurs and investors who really have that same vision that we share as well, especially for the US market, where I think there's a huge opportunity for this. Otherwise, yeah, I'd be happy to entertain any questions or comments, and look forward to your feedback.